talk, we're going to speak about the requirements for the growth of plants. In flowering plants, growth resides on food material. But in almost every plant, at some stage in the life cycle is bridged by a single cell. By spores, which are single cells that can grow, or by gametes, which are sex cells, and these only grow when they fuse. But now let us turn to the reproduction of a flowering plant, a typical flowering plant as illustrated in the case of the cherry, as shown on this chart. The egg is contained in the embryo sac. The embryo sac is itself in a structure called the ovule, a sort of a nursing organ. The actual process of fertilization of the egg occurs when one male cell, one male nucleus, fuses with the egg. And this eventually forms the zygote of the embryo. There is a stimulus to grow occurs when this process of fertilization occurs. But there's a second stimulus to grow. When the second male nucleus fuses with this nucleus here, and this growth outpaces at first that of the embryo. But it produces first a sort of liquid structure and then a cellular structure which we know as the endosperm. And in the endosperm is deposited the food material for the growth of the embryo and the nucleus, and not only the food material, but the stimuli, the cell division stimuli which are needed at this stage to make these various nutrients and stimuli available to the embryo. It sends out cotyledons which absorb this material from the endosperm. So now let's turn to another chart which will tell us more about these cotyledons. Here in the development of the seed of a shepherd's purse, you see the cotyledons going down into the endosperm, absorbing the material for the young embryo. If we cut a slice across a grain of buckwheat, we see a massive amount of starch and the little thin white papery cotyledons which will absorb this material when it grows. But now let's turn to a very special and a very useful case, namely the coconut. This very large structure which has in it a large amount of liquid, the so-called liquid endosperm. Here we see the nut in its outer husk, which falls on the ground. The embryo is immature, but when it grows, this sends out a structure which is a cotyledon into the center of the nut. This absorbs the whole of the liquid and nourishes the young embryo. So the cotyledons are the absorbing organs, which absorb not only the food, but the special stimuli, the cell division, which the young embryo needs at this point. Now let us imagine then that the young embryo has established its shoot growing point and a root growing point. And let us look at the structure of the shoot apex as represented by a longitudinal section through the shoot apex of a common mint plant. In the longitudinal section of the mint plant, we should note the following. First, the tiny, rounded, central apical dome. Secondly, the abundant cell divisions on its flanks, which give rise to the overarching leaves. Thirdly, the little pockets of dividing cells, the axils of the leaves, which give rise to buds. And finally, we should note the strands of dividing cells, which remain dividing after cells on each side have ceased to divide. These are the procambial strands that give rise to the cambium, which is the tissue that makes lateral growth in girth of the plant body. This tissue is able to continue to divide when other cells have ceased to do so. In fact, in a sequoia tree, it may be able to divide for 4,000 years, although with alternating periods of activity and rest. We see then that we need to be able to understand what are the factors that determine whether these cells grow by division or remain alive without dividing. So now let's turn to the question of the requirements for this process, some of which come from the external world requirements come from the leaves. In this chart, we see an abbreviation of a chart which was published many years ago in a book called Hunger Signs in Crops. It tells you the requirement for an acre of corn in terms of the water, the oxygen, the carbon, the nitrogen, and so forth, which is needed. And we see over here, in pounds per acre, the amount of these various materials. Just to make these figures meaningful, let's take note of the fact that this amount of water represents 19 to 24 inches of rain. This amount of carbon represents 4 tons, the equivalent of 4 tons of coal. This amount of nitrogen is the equivalent of 8 100-pound bags of 20% nitrogen fertilizer, and so on. The amount of calcium represents 80 pounds of limestone. But when we come to these materials that are needed in much smaller amounts, the units are very different. The amount of iron here is the equivalent of about 2 pounds of nails. The amount of manganese is the equivalent of 1 pound of potassium permanganate. The amount of boron, which is needed only in very small amounts, could be supplied by a quarter of a pound box of ordinary common borax. And the chlorine, enough of this, would be supplied in the rainfall alone. The zinc, by the shell of 1 dry battery. And enough copper would be furnished by 25 feet of very thin copper wire. But there's one element which is missing. It was not known when this chart was originally drawn up. We now know that the element molybdenum is needed, and that a very tiny amount of this is enough for the growth of an acre of corn. But now we know that these nutrients are furnished in solution. And the interesting thing is that the plant is able to receive its nutrients in very dilute solution, because it's able, as we say, to accumulate ions, to make the ions move from a dilute external solution into a strong internal solution, to make them, as it were, move uphill against an adverse concentration gradient. The plant Nigella, which grows in a pond, has been useful in this connection, because investigators have analyzed its sap and compared it with the external solution. And this ratio, the so-called accumulation ratio, shows that the Nigella can multiply the concentration of sodium and potassium and chloride very greatly in its sap from that which occurs in the outside solution. A plant which grows in the sea, Valonia, here we see 2 species, Valonia macrophysa and ventricosa, also does this. Whilst it keeps out sodium, which ratio is less than 1, it accumulates potassium many, many times. Although Nigella and Valonia were especially useful in establishing the fact of the accumulation of ions by plants, other plant materials proved more useful in showing the nature of the process. Here on this chart we see an example of work done with thin slices of potato tuber, which were placed in water and aerated with a gas mixture containing different concentrations of oxygen. On this axis we see figures which represent the relative amounts of the bromide ion, which were accumulated by these discs of potato tuber. And as can be seen, there's a very sensitive relationship between oxygen concentration in the solution and the bromide ion absorbed, with a critical oxygen concentration at or about that which obtains in air. Had we shown on the same chart the effect on the respiration of the potato discs, we should have seen that the respiration was affected in somewhat a similar fashion, showing that respiration is intimately involved in the machinery of taking the ions from the dilute solution outside into the vacuole solution of the potato discs of these now essentially growing cells. But we can also illustrate the same idea if we turn to another set of data taken with another kind of system. In this case, the experimental material were the roots of barley plants, seedling barley plants from which the shoots were excised. This work was done at the University of California by Hoagland and Breuer. Again, the roots were exposed to a solution which contained different oxygen concentrations, as shown by the percentage of the gas which was bubbled through the solution. On this axis, we see the effect on the absorption of the potassium ion, the nitrate ion, the chloride ion, and also, along with this, the actual loss of sugar, which accompanies the respiration that occurs. So here again we see that the living cells are able to use the vital energies, the vital machinery that they possess to bring about this movement of ions from a dilute solution outside into their own vacuole. However, we should note that the living cells contain in their internal fluids much more than salts. For there are other solutes essential to growth which come from other sources. There are sugars which come from the leaves, organic nitrogen compounds which are metabolized, are made by the cells from, for example, from nitrate which is absorbed from the solution and sugar. There is in fact a great diversity of amino acids in such cells. And by the modern techniques of paper chromatography, we can now spread out these amino acids on a sheet of paper, stain them with a reagent, an anhydrin reagent, which reveals them, and show their identity. And in fact, in the illustration, we can see, spread out for the eye to see, the amino acids of the potato tuber appropriately named. And these are the substances that furnish the nitrogen, which is the raw material for the synthesis of protein in these when the cells begin to grow again. But even with all these solutes present in the cells, salts from the environment, organic compounds from the leaves, etc., there are still other factors which determine whether or not the cells will grow. In fact, the cells of a potato tuber may seem to have all the nutrients already there, which are necessary for their growth. But what is it that triggers off the growth when it finally occurs? Here we turn to the technique of tissue culture. For this is a technique which has told us much about the accessory growth substances which are necessary to trigger off the growth over and above the mere effects of the nutrients. At the end of the last century, a great German botanist, Haberland, saw how useful it would be if we could take cells of the plant body and culture them outside in a completely artificial medium. Since then, by the work of Robbins and White and Gauthier and many others, we've learned a great deal about this problem. But there's still a great deal to know. We need to know, for example, why it is that the cells of the cambial tissue will grow readily, whereas other cells require additional stimuli. We need also to know why it is that in some cases the process seems to go wrong, and cells which normally would be quiescent begin to grow again and may even form a tumor. For example, when the crown gall organism attacks a palenquid plant, a tumor may be seen to form. We now need to distinguish the different ways in which cells grow. So they grow by cell division, that is, they multiply, and they grow by cell enlargement. Until recently, much more was known about the accessory substances that control growth by cell enlargement than about the substances that control growth by cell division. But in any event, much of the terminology in this field was inherited from work with animal systems. and such terms as vitamins and hormones are more difficult in their application to plants than they are to animals. For example, a vitamin is characteristically a substance that is produced outside the organism which it affects, whereas the hormone is produced in one part of the body and acts, as it were, at a distance. Thus we see, however, that in plants, the vitamin thiamine, for example, affects the growth of roots, although it may be formed in leaves. For the leaf is perhaps the most important vitamin factor in the plant body. Characteristically, the substance indole acetic acid is known as a hormone, and by the work initiated by Darwin in the last century, followed by many other investigators on the strange organ known as the coleoptile of grasses, we now know much about this substance. It's now necessary then to make a brief summary of the work which has been done by such people as Wendt and Thiemann and Kerbel, leading to our knowledge of the role of indole acetic acid in the growth by cell enlargement of plant cells. These charts show in very summarized form the history of the main events which have led us to our present understanding of the role which auxine or indole acetic acid plays in the growth of cells by enlargement. As we can see, Darwin here showed, 1880, that if the tip of the coleoptile of grasses is intact, it allowed this organ to bend toward the light. But if the tip was interrupted by a barrier, then flow of active substance was impeded. If the tip was removed and replaced, some activity moved back from the tip to allow the stump to extend. Again, if the tip is placed asymmetrically to one side of the organ, the resultant curvature is interpretable on the assumption that the tip activated the side above which it was placed. The activity also can pass through a permeable barrier. And this all led to the very interesting experiments by which Wendt was able to concentrate the substance from the little tips of coleoptiles into gelatine blocks, which could act like the tip itself when these were placed on coleoptile stumps. All this work, with much very delicate chemical work, has led to the understanding that at least one of the active substances that produces growth by cell enlargement is the substance indole acetic acid, which is known as an auxine. This, however, is all dealing with growth of cells by enlargement. If now we turn, however, to the stimuli that cause cells to grow by cell division, a different set of circumstances needs to be considered. Here we see what happens if we take little tiny explants or plugs of carrot tissue from an ordinary carrot root, and under appropriate circumstances, that will be described later, we place them in a medium, otherwise complete with all the nutrients and salts and vitamins that the carrot should need, but supplemented now with the fluid obtained from the central cavity of the coconut, the liquid endosperm or coconut milk. Under these circumstances, the carrot grows rapidly and eventually will increase its weight as much as 80 times in about 20 days. But this is increase in weight alone. This is mainly increase in water. What are the cells doing? If by appropriate means we macerate this tissue, break it down into individual cells, and then by special means count these cells, and we find that the original explant that we removed had in it about 25,000 cells. But in response to the coconut milk stimulus, the number increases to something of the order of 1 million, in something less than 20 days. And this rate of increase could continue. Thus, the cells of the carrot, which had originally stopped growing, treated with the nutritive fluid of the coconut milk, return again to the state of active cell division. The question now arises, how were these results obtained? The film you are now about to see was taken in the Cornell laboratories, and the investigators and the assistants who play part in this work you will see in the film. The little pieces of carrot tissue are removed from the carrot root with all the surgical precautions that are adopted in an operation, because everything has to be sterile. The little pieces of tissue removed from a distance from the cambium such that they would not otherwise grow, are placed first in sterile water, and then removed from the dishes of sterile water into special tubes with a central side neck closed with a cotton plug. Into the tube is placed some nutrient medium, 10 cc or 10 ml, we often say perhaps, containing the nutrients of a basal medium containing salts and vitamins. With or without the coconut milk or other cell division promoting extracts at will. As the tubes revolve around the horizontal shaft, a little piece of tissue is exposed alternately to air or to water. And it is under these conditions that it grows, stimulated by coconut milk in the way that you have seen on the preceding growth curve. But also, we can grow this tissue in other ways, because in the film and on the rotating shafts, we see some special flasks. These were first devised to grow the tissue in bulk, but under the conditions in which a large number of carrot explants were placed in these curious flasks with their little side necks, their little nipples around the middle of the flask, we found that not only did the tissue grow well, but there were free-floating cells in the medium, and these also showed signs of division. Thus we have a technique here by which we can at will turn on the processes of growth by cell division or allow it to remain shut off. Not all cells, however, will grow stimulated by coconut milk alone. For to grow cells of the potato tissue, we need an additional chemical stimulant. And the first example of this that was known was the substance more familiar as a weed killer, namely 2,4-D. This is one of many examples now known of substances known as synergists, which combine with the coconut milk to make the cells grow by cell division. Thus we have here, then, a powerful example of the way in which the nutritive fluid of the coconut restores to otherwise resting cells the kind of divisions that we were familiar with at the outset of the talk in the egg as it occurred in the embryo sac of the ovule. There is, however, another important difference between the behavior of the free cells and the explanted piece of carrot tissue. The preformed tissue grown in the presence of coconut milk never organizes to form roots. But the free cells, as has been shown, will grow first into clusters, then form roots and shoots, and eventually whole plants with normal flowers. Thus the coconut milk, acting on the cells free from their neighbors, seems to superimpose the ability on these cells to behave again like zygotes or as embryos. This can be shown dramatically in these two sets of charts. Here we have the normal embryology of the carrot, from egg to young embryo. Here we have some cell clusters of cultured cells which resemble this sequence in a striking fashion. But in addition, if we go to free cells of a plant like Haplopappus, which only has 4 chromosomes, we find that these adult cells will divide quite frequently in such a way that the original 4 chromosomes are reduced to 2. And in this way, the essential reproductive step of reduction division has been obtained in otherwise mature adult cells. Work on the chemical controls that determine growth of cells by division owes much to work on coconut milk, on extracts of immature corn, and on extracts obtained from horse chestnut fruits. In all of these materials, these three substances, or similar ones, have been isolated from what we call a neutral fraction. This one even obtained in the amount of 200 grams from 20 liters of coconut milk. But these substances only create the conditions conducive to the action of the growth factors proper. And it would be far too complicated a subject to deal with this fully here. However, we know, as in the chart that you can see on the screen, that many substances now need to be considered in this way. Substances that promote growth by cell division, substances that promote growth by cell enlargement, and their inhibitors. But the end is not yet in sight. In the understanding of the chemical controls that determine how the built-in capacity of a cell to grow is unleashed, much yet needs to be done. And as we approach the day when we understand how the totipotency of a whole mature carrot cell can become a whole carrot plant, we may expect to know more about the growth of all organisms, from bacteria to man. Attention will be focused upon the ways in which the built-in capacity to grow in the embryo unfolds. For this aspect of the general problem, the terms development and morphogenesis are often appropriate. For the most part, we will speak about flowering plants, but equally important lessons could be learned from other parts of the plant kingdom. The topic of growth usually begins with a seed. But prior to that, quite important and quite long and important stages of development occur in the formation of an embryo from a fertilized egg, in the formation of a seed from an ovule, in the formation of a fruit from an ovary. Commonly, there is stored in seed, often in the cotyledons, as in this legule here, much stored food material. In some structures which grow, like a grain of wheat or a grain of corn, the food material is stored for the young seedling, for the young embryo, in the endosperm. Some structures, which are often confused as seeds, are really fruits. And in the case of these buckwheat grains, this is the case. And a lot of stored food material is in the endosperm, as also in the endosperm of these castor beans. But not all seeds are large, and not all seeds are endowed with this large amount of stored food material. But having got the seed with its stored food material, the events of germination are triggered off by the absorption of water. It's a familiar fact that in the absorption of water and in the growth of even very delicate plants, great forces are exerted. Mushrooms can lift paving stones. However, we can demonstrate the forces which are generated in the absorption of water during the germination of some seeds by this demonstration with mustard seed. Here we have an ordinary medicine bottle packed full with mustard seeds. Water has been placed in the bottle to displace all the air. This is very important. If we now, however, finish off this and pour water in the remaining space so that the bottle is completely full to overflowing with water and mustard seeds, and then we will close it with a stopper really tight, get it really tight, and place it over here. We will now leave the bottle for about 40 minutes, and as the seeds swell, the bottle will break. However, food and water are not all that is involved in the germination of seeds. Some seeds will not germinate for long periods, even though they're under otherwise favorable conditions. There's a problem then to know what determines the period of rest and dormancy. Some seeds have hard seed coats. Some seeds require exposure to low temperature. In some seeds there are chemical inhibitors which determine whether or not the seed will germinate. All this is too complicated and too extensive a subject to be dealt with fully here. But a dramatic example of this is now being studied in the U.S. Department of Agriculture laboratories at Beltsville. And I have here some pictures which show what is being done with the germination of lettuce seed. In this particular picture, some lettuce seeds in a dish have been exposed to darkness, and few of these germinate. But when exposed to red light, the seeds germinate almost completely. So the germination of these seeds is determined by their alternate exposure to darkness or to red light, as the case may be. In this dish, the seeds have been treated with different periods of exposure to red light. The longer the period of exposure to red light, the more complete the germination. So we can see that the process of germination is determined by a variety of quite complex conditions. In the germination of the seedling, the root or radicle usually emerges first, and the little shoot or plumule follows later. But in the formation of the seed, the organization of the shoot and the root is determined at a very early stage. As we say, polarity is developed, because these growing regions respond in a quite different way to stimuli. We can see something of the organization of the shoot growing point by looking at the growing point of a mint plant. We shall see this in section also. But as we see on this shoot, the leaves are laid down in what the botanists call opposite and decussate fashion, because the two little lobes which are to become the leaves are formed in the apex close to the central dome of the growing region. They then grow up and overarching the central apex. In this opposite and decussate type of leaf arrangement, the first pair of leaves are opposite each other, and the next pair of leaves, also opposite, are between the gaps between the... This looks as though each new site of leaf formation is as far away as possible from its predecessor. However, in this kind of opposite and discussive leaf arrangement, sometimes anomalies occur, as can be seen in this photograph of an anomalous mint shoot. In this shoot, instead of having 2 leaves at each node, there were 3. And at the node below, 3 other leaves form opposite and form below the gaps in the leaves, between the leaves above. So this is a very common phenomenon in plants of this sort, and it looks as though on a rather larger shoot apex than normal, more leaves can be accommodated at a given level. By contrast, the apex of the root is quite different. As the section shows, it is free from the lobes which in the shoot form the leaves, because the lateral roots form deep-seated, far from the surface of the root. If we examine the extreme tip of the root, right in the center where one might expect to see the most actively dividing cells, there are some rather large cells, which obviously do not divide as frequently. This is the so-called quiescent region. But radiating out from these larger cells are files which seem to show that the initiating cells could act as sources of the stimuli that determine the division and development of the cells behind. However, we now need to ask, what is the behavior of these shoot tips and root tips to external stimuli? There are 2 problems. What determines the direction of growth which obtains, and what determines the kind of structures to which the shoot particularly gives rise? This is the great area of plant physiology which has produced a lot of the knowledge about hormones for this attracted early attention. The main facts are relatively simple to state. Shoots grow upward, roots grow downward. Whatever the orientation of the grain of corn in this case, or of another type of seedling, whatever its orientation to gravity, the shoot quickly grows up and the root goes down. The root is positively geotropic, the shoot is negatively geotropic. If we place a plant in a horizontal position, its shoot grows up, and if we could see its root, it would probably grow down. However, there is also a response to light. In this black box, a narrow slit allows the seedlings that are in the container inside to be oriented unilaterally to light. These are little mustard seedlings. And if I lift them out, you will probably see that the shoots are turning all toward the light. Were you close enough to see this, some of the roots would grow away from the light. For in this plant, rather rarely, both shoots and roots are sensitive to light. However, we now know that in the tip of the shoot or the tip of the root, this stimulus to light or gravity is determined by the movement of activating substances to the so-called motor region in question. So this response is brought about by elongating cells in the direction of maximum growth by elongation. It is this area of plant physiology which has produced our knowledge of the hormones of which indole acetic acid or auxine is a prime example. It was the organ known as the coleoptile, studied first by Darwin, which produced this information originally. But we also know that other substances, other classes of substances, produce somewhat similar effects. Because the class of substances now known as gibberellins, which are able to convert dwarf plants into tall plants, these also act by determining the elongation of cells. But there are many other areas of response of plants to environmental stimuli. Many of these involve light. And because they involve the form of the plant body, the appropriate term is morphogenesis. Shade leaves and sun leaves differ in size and shape. Juvenile leaves are often different from adult leaves. Submerged leaves of water plants may differ from the aerial leaves. But a very striking response is that induced in plants by growth in complete darkness, or what we call physiologists. In this black box are plants that have been grown in complete darkness. In this case, some legumes, which show this effect very strikingly. If I remove the plant from darkness, you'll see that the stems look more like roots, but they have this very characteristic curvature known as the plumula hook. If we examine the anatomy of these stems, they will be quite abnormal. In order to see these effects, we have to treat the plants with the same care that we would photographic plates. Because on exposure to light, they quickly respond, the hook straightens up, new leaves begin to form. But this type of response has been studied in order to understand more about what the light does. At Beltsville, in the laboratories of the U.S. Department of Agriculture, is a very large spectrum which has enabled the effect of light to be related to its wavelength. This has given rise to the idea that in these so-called etiolated plants there is a pigment called phytochrome which absorbs the light and enables it to trigger off the response in the growing region. But it is this area of plant physiology which is now yielding a great deal of information on the stimuli, environmental and otherwise, that activate the growing regions to produce the kind of growth which occurs. The most fascinating response of all, which occurs in the shoot, is perhaps that which determines the transition from a vegetative to a flowering plant. Plants do this in many different ways. Some, like the chickweed, can form flowers in a few weeks. Some only do so, like many perennials, after many years. Some plants are biennials and require the stimulus of a winter rest and exposure to cold. Some, as we shall see, require a special stimulus, the photoperiodic stimulus, which is determined by length of day, or perhaps more precisely, length of night. Sometimes biennial plants inconsiderately form flowers in their first season of growth. This is often true of some biennial crop plants. And here we see a picture of some celery plants exposed to low temperature in the seedling state. This type of response was discovered in the 1920s by Professor Thompson, then working at Cornell University. But as we turn now to this stimulus of length of day, or the photoperiodic stimulus, we here see a very great deal of information summarized in a chart, for which, incidentally, we are indebted to Professor Lang from the California Institute of Technology. This refers to a plant, the cockleberry, which has been greatly used in this kind of work. And as one can see, if a short night after a long day If the plant is exposed to a short night after a long day, it remains vegetative. If it's exposed to a long night after a short day, it forms flowers and fruit. A long night, however, may be interrupted by a brief period of exposure to low night intensity. Without a very long night may be exposed by successive periods of exposure to light of low intensity. And under these latter conditions, the plant remains vegetative. It is this kind of response, then, which has produced so much interest in these photoperiodic effects, which are thought perhaps to be determined by the phytochrome pigment to which reference was previously made. Sometimes, however, the temperature effects and the photoperiodic effects interact. For example, a mint plant is normally a long day plant, but if grown at low night temperature, it behaves like a short day plant. Similarly, cabbage plants are normally biennials, but if grown at persistent high temperatures day and night, they may be kept vegetative for many years. Thus, it is this kind of interaction between the environmental conditions and the growth and behavior of the plant that has produced at the present time so much interest in the growing of plants under standardized conditions. A pioneer laboratory, the Earhart Laboratory, under Professor Wenck's guidance at the California Institute of Technology, was devised for this sort of study. And since Professor Wenck is now operating a new venture of this sort in the bioclimatic laboratory at St. Louis, it is appropriate that he should continue this discussion and tell more about this sort of development.
Thank you, Dr. Steward. Environment is an important factor in the development of all organisms, not only because they respond to it, but also because they are shaped by it. Now, what is environment to a plant? That is the air in which it lives, the soil in which the roots grow. It is the insects and the diseases which attack it, or the grazing animals. Now, since this environment is so complex, how can we find out about what it does to a plant? Well, we can do this by completely controlling the environment, which is possible more recently because of two developments. In the first place, because we have now the possibility of air conditioning greenhouses and artificially light rooms. We can then maintain the proper temperature, the proper humidity, and the proper wind conditions for plants. And in the second place, because of the fluorescent tubes which give just the right light intensity and the right type of light for the growing of plants. The intensity for a normal growth of plants is very high, and it has to be something like 50 times as high as what you have in your classroom. This is then possible now with these new lights, which in the rooms where they are used, just about plaster the whole ceiling of the room. Let's now consider specifically what the effect of temperature is on the growth of plants. The temperature, of course, differs very much from one place to another place in the world, and in a controlled temperature laboratory, let's see what the results are. I have here a picture which shows the response of a particular plant, the Coleus. All the plants in this picture are of the same age. They only were kept at different temperatures. In the upper row, you see a whole set of plants which all were grown at a day temperature of 72 degrees Fahrenheit. Whereas the night temperature varied from low at the left to high at the right. And you see there that at one particular temperature, at 73 degrees, at 62 degrees Fahrenheit night temperature, that we have the best growth. That's what we call the optimal temperature. And when the temperature rises beyond that, then growth decreases. In the next row, just below it, you see the same type of plant, but here it is grown during during day. So here we have a plant of which we know exactly what the optimal temperatures are during day and night. Now, the most modern greenhouse in which we can control pretty accurately both day and night temperature is the climatome, which just has been built at the Missouri Botanical Garden, and of which you see here a picture. As we enter the climatome, we are immediately struck by the very fresh atmosphere which is there, even though it is warm, because we are in a tropical climate. Actually, we are in several different tropical climates because we are maintaining in the climatome temperature differentials from one end to the other. So in the first area where we are now, we see an Amazonian jungle. This is a very rich experience with lots and lots of greenery plants growing all around. We actually don't see too many flowers there, but this is a real reproduction of the jungle as it's growing. Normally, a jungle, once it is cut, does not develop in the same way as it was. We first get some very rapidly growing plants, and only in the shade of them are the slower growing jungle giants coming up. These are seen here. For instance, the Hymenea courbaril, which grows in the shade of the fast-growing ant plant Krikorian, and the particularly fast-growing balsa wood of Orma latifus. You see how this has grown during the last 9 months, almost 20 feet. As we go down the lower level of the climatome, we come to a jungle of the highlands of the tropics. This is a waterfall which is completely surrounded by the aeon lily family objectives, and we have bananas, relatives, growing with large shiny leaves. Among the more remarkable flowers which we see are the passion flower. This is a most beautiful flower, very intricate, and either maybe blue or in other species red. The orchids are usually small, but there are some very big ones, as for instance this moth orchid, which grows as an epiphyte in trees. An epiphyte is a plant which uses the branches of trees just as a support and is able to get a sufficient amount of nutrient from the rainwater which runs down the branches. Another epiphyte which you see here is a bromeliad, which is a relative of the pineapple. This has very heavy, stiff leaves, of which the bases are so closely knitted together that rainwater will stay in them, which is a good supply of water in between rains for this plant. The Mimosa pudica, the second name meaning bashful, because it closes its leaves upon touch, is shown here with delicate pink flowers. Then it is approached by a flame, the leaflets immediately start to close on the leaf, then the whole leaf will drop down, and the stimulus will then further travel down and up the main stem. In just a few seconds, you see the first lower leaf drop down, then the next higher leaf, and then again the second lower and the second highest leaf. In this way, the stimulus can be actually followed from place to place in the plant by the movements. Another plant which shows movements is a telegraph plant, Desmodium gians, which is seen in this picture. It has compound leaves, of which the two small side leaflets are in continuous movement and are waving back and forth just as if they are signaling each other. The different climates which we have in the climatome are also being used for research. We can, for instance, grow corn in the different areas, and the corn actually shows big differences in growth according to the temperature where they are grown. In the coolest temperature, the corn, for instance, had hardly grown to 6 feet height, whereas in the warmest conditions, it grew up to 18 feet. When there is so much machinery necessary to keep the climate home under proper temperature conditions and climatic conditions. You see switches and automatic machinery to control fans, heaters, etc. Every 3 nights out of the week, we have artificial lights in the climate home, and it is open to the public. This would give great difficulties with plants if we left them on every night, because then they would be kept under long day conditions the whole year round, and plants would either flower or not flower at all, as you have heard just from what Dr. Stewart told about the effect of photoperiod on plants. In conclusion, we can then say that we'll find more and more of these climate-controlled greenhouses in different places in the world for the education and information of the public, and for further research on plants